Hello, I didn't see you there. Welcome to this week's RAM course on how glucose goes through a lipid bilayer. How about, let's get started.
Oh, hi, I'm Sam. So, today on RAM course, in order to get through our problem, which is to get the glucose through the lipid bilayer, we must use a quaternary protein. In order to do this, we have to go through the protein structure. There are four structures: primary structure, secondary structure, tertiary structure, and quaternary structure. Hi, welcome back. Right now we are going to be talking about dehydration synthesis. Dehydration synthesis goes with bonding and getting two monosaccharides into one disaccharide by losing H2O. Proteins form a long chain of amino acids. Amino acids determine the structure and function of proteins. There are four different types of amino acids. There are basic, polar, acidic, and nonpolar. In order to carry out the function the protein must do, it has to form into a new shape. Primary structure, the first structure, looks like this. These little things are the amino acids. The primary structure is a linear shape.
Oh, hello there. I'm Deja. The next step after the primary structure is the secondary structure. In the secondary structure, there are two different shapes the polypeptide chain can form. One of the forms of the chain can take is the alpha helix. Another is the beta-pleated sheet. Both formations are determined by the amino acids in the primary structure. The folds in the polypeptide chains are created and then supported by hydrogen bonds. The H from the amino group and the O from the carboxyl group.
Hello there. I am live here in the redwood forest to tell you about the tertiary structure. The tertiary structure is simple to understand. I have an example here. It is a long chain of alpha helixes and beta pleated sheets coming together and forming a completely different shape.
Oh, hello. How are you doing today? Well, let's continue our next segment of quaternary structures. What are quaternary structures? What are they made of? How do they work? All fantastic questions. Let's jump right into it. A quaternary structure is made up of tertiary structures, as we know contain alpha helixes, beta-pleated sheets, and long strains of polypeptide amino acids. Now, they are bonded together due to something called disulfide bridges, and they can come up with two different structures. One is fibrous structure, and the other one is known as globular structure. Now, a fibrous structure is much more fibery-like due to the name fibrous structure, and a globular structure is much more globular due to the name as globular structure. Now, when the tertiary structures bond together, the number doesn't matter. It could be 2 bond together due to disulfide bridges, or 2,000. The number does not matter, as long as they bond together due to disulfide bridges. Thank you for your time.
Welcome back to RAM course. Now we are going to be solving the problem that we were talking about earlier. How to get glucose through the quaternary protein. Okay, so in order to get through glucose through the quaternary protein, we have to go through the lipid bilayer. Lipid bilayer has a phosphate group, a glycerol, and fatty acids. At the end of the fatty acids are stage 3, which is methyl, which means that the protein has to be nonpolar on the outside because methyl is nonpolar. So the functional group that we will be using on the outside of the protein is methyl group. Then on the inside we will need something polar because glucose is polar. So we will be using hydroxyl on the inside. So now that we have the nonpolar and polar, glucose can go through the protein safely.
Yeah! High five, guys! What in the world's gone over here? Go back to class! Learn!
No one told you life was gonna be this way. Your job's a joke.